turn off your device camera. Participants are muted by default to enable the speakers to present without interruption. Participants can ask questions by visiting slido.com using the code Bioinfolecture or click the link in the chat box. To ask your questions, state your name, affiliation, and the question you want to ask. You can also upvote questions already posted by other participants. To get your certificate of attendance from this webinar, fill out the evaluation form. The link will be posted in the chat box at the end of the webinar.
The coronavirus nationwide lockdown has left the streets nearly deserted. People have been ordered to stay home. Get back to what you used to do. We must set up several layers of barriers against the virus, but each barrier is not 100% effective. So to be fully protected, all barriers have to be in place. One of the key barriers is proper hygiene. Another important barrier is setting a distance between you and others who carry the virus. The other way around is also true. Protect the others from your germs. The government has also taken measures to protect the public through testing and contact tracing, quarantine and isolation, and finally, the last and most important barrier, vaccination. PGC Mindanao has been actively exploring ways to help the local efforts against COVID-19. At the end of the day, we just want to get back on our feet. Vaccination is the final barrier that completes your protection against COVID-19. So why settle for less? Be vaccinated.
Please turn off your device camera. Participants are muted by default to enable the speakers to present without interruption. Participants can ask questions by visiting slido.com using the code Bioinfolecture or click the link in the chat box. To ask your questions, state your name, affiliation, and the question you want to ask. You can also upvote questions already posted by other participants. To get your certificate of attendance from this webinar, fill out the evaluation form. The link will be posted in the chat box at the end of the webinar.
The coronavirus nationwide lockdown has left the streets nearly deserted. People have been ordered to stay home. Get back to what you used to do. We must set up several layers of barriers against the virus, but each barrier is not 100% effective. So to be fully protected, all barriers have to be in place. One of the key barriers is proper hygiene. Another important barrier is setting a distance between you and others who carry the virus. The other way around is also true. Protect the others from your germs. The government has also taken measures to protect the public through testing and contact tracing, quarantine and isolation, and finally, the last and most important barrier, vaccination. PGC Mindanao has been actively exploring ways to help the local efforts against COVID-19. At the end of the day, we just want to get back on our feet. Vaccination is the final barrier that completes your protection against COVID-19. So why settle for less? Be vaccinated.
Please turn off your device camera. Participants are muted by default to enable the speakers to present without interruption. Participants can ask questions by visiting slido.com using the code Bioinfolecture or click the link in the chat box. To ask your questions, state your name, affiliation, and the question you want to ask. You can also upvote questions already posted by other participants. To get your certificate of attendance from this webinar, fill out the evaluation form. The link will be posted in the chat box at the end of the webinar.
The coronavirus nationwide lockdown has left the streets nearly deserted. People have been ordered to stay home. Get back to what you used to do. We must set up several layers of barriers against the virus, but each barrier is not 100% effective. So to be fully protected, all barriers have to be in place. One of the key barriers is proper hygiene. Another important barrier is setting a distance between you and others who carry the virus. The other way around is also true. Protect the others from your germs. The government has also taken measures to protect the public through testing and contact tracing, quarantine and isolation, and finally, the last and most important barrier, vaccination. PGC Mindanao has been actively exploring ways to help the local efforts against COVID-19. At the end of the day, we just want to get back on our feet. Vaccination is the final barrier that completes your protection against COVID-19. So why settle for less? Be vaccinated.
Please turn off your device camera. Participants are muted by default to enable the speakers to present without interruption. Participants can ask questions by visiting slido.com using the code Bioinfolecture or click the link in the chat box. To ask your questions, state your name, affiliation, and the question you want to ask. You can also upvote questions already posted by other participants. To get your certificate of attendance from this webinar, fill out the evaluation form. The link will be posted in the chat box at the end of the webinar.
Tayo po ay magsitayo. Bilang pagpupugay, ilagay ang kanang kamay sa ating dibdib at awitin ang pambansang awit ng Pilipinas.
Ladies and gentlemen, your host, Dr. Richie May Gamont.
Good afternoon, everybody. Thank you for taking time to join this webinar. And on behalf of PGC Mindanao, welcome to the very first Bioinformatics lecture series. I am Richie May Gamont, and I will be your host for today. This lecture series is a collaboration between the Research and Development Division and the Bioinformatics Division of PGC Mindanao. This event will bring you exciting bioinformatics topics as applied to diverse fields. Today, we start with our very own PGC Mindanao research staff, who will speak on a topic as applied to agriculture. Next month, we will also be featuring two leading UK academics from the University College London and University of Birmingham, who will be sharing with us UK efforts in contributing to studies related to COVID-19 and in bioimage analysis. To start off with this bioinformatics lecture series, let me introduce the moderator for this session. Our moderator is currently a University Research Associate at the Department of Biological Sciences and Environmental Studies in UP Mindanao. She is also assigned to the Omics Laboratory Service Division of PGC Mindanao. She completed her master's in molecular biology and biotechnology at the University of the Philippines, Los Baños, and conducted her thesis at the International Rice Research Institute as an affiliate master's research scholar. Her research interests include tissue culture, biodiversity, and genetic engineering. Everyone, please help me welcome Ms. Joan T. Acaso.
Thank you, Dr. Chimi, for that kind introduction. And of course, I would also like to thank all our 500 plus participants from different parts of the world who are here with us in this afternoon's lecture. Maraming salamat po. Okay, let's get ready. I will be introducing our first speaker for this lecture series. So he graduated with his BS in biology, major in zoology from the Mindanao State University Iligan Institute of Technology. Shortly thereafter, he proceeded to take his MS in molecular biology and biotechnology, MBB, with cognate in biochemistry at the University of the Philippines, Los Baños, and he is also an affiliate scholar at the International Rice Research Institute during his MS, where he focused on exploring downstream bioinformatics analysis pipeline via genome-wide association studies, or GWAS. This is to accelerate novel gene discovery in rice against blast pathogens. He worked briefly in Erie, juggled between C4 Rice project and Erie's bioinformatics team, where he primarily handled the wild rice mitochondrial genome in an effort to identify mitochondrial genes that could be utilized in breeding program to supercharge rice photosynthetic machineries, thus increasing yield. He's also currently a PhD student in UPLB, taking up agricultural chemistry with double cognate in MBB and agronomy. And he will be working on the rapid detections of cacao's fungal pathogens using functionalized magnetic nanoparticles, or MNPs. Yes, without much ado, let us welcome in our virtual screen Mr. John B. DIVINA, our current university researcher, for his lecture. A warm virtual of applause, please.
All right, thank you, Joan, for that comprehensive introductions. And to all our participants in this first Bioinformatics lecture series, I am glad that you were able to make it into this webinar, and I hope that you are all in safe comfort of your family and loved ones. For this webinar, I will share some insights on one of the area that we in PGC Mindanao are currently looking into, that is on the applications of genomics to solve pressing on pressing agricultural problems. In the interest of time, I decided to prepare a recorded presentations for this webinar, and this is also to ensure that I will be able to parse my presentations into the allotted time. I will be live again, of course, to answer your questions for questions during the Q&A sessions. And finally, I hope that to our participants will be able to learn a couple of things out of this webinar, which will be also useful in the research endeavor. And with that, I think our technical team is already okay. So I give the virtual floor to our technical team to share the recorded video. Thank you.
So good afternoon again to everyone. As I said earlier, for today's afternoon, I will be talking on the two fields that have been long reinforcing each other and to some point share values that has led to the development of agricultural genomics and its useful tools and approaches. Now, my talk will be structured into four main parts. The first one is on agriculture, where I will walk you through the global agriculture trend and local agriculture trend. I have to say that I will be biased towards crops, but approaches in crops can also be applied to animals. The second is on genomics, where I will highlight some key development of high-impact genomics platform. Now, the third naturally developed as genomics were applied to solve key agricultural problems, which then lead to the development of tools as the result of this crosstalk. Now, it would be worthwhile to spare a few moments to highlight this man, Norman Borlaug, who is also known as the father of green revolutions. Now, his contributions is immense. With the assistance from Rockefeller and Ford Foundations, of course, he was able to develop a successive generations of high-yielding wheat varieties with broad and stable disease resistance. And he was also able to develop a variety wheat varieties with broad adaptations to growing conditions across diverse environments. Now, this led to a remarkable increase in wheat productions and also laid the foundations for the subsequent rice production. Now, the Green Revolutions corresponds to the period between 1960 and 1980, where there is a remarkable increase in the productions of wheat and rice. It was achieved after a period of considerable decline, as shown by the graph portions in circle green. And the increase in wheat and rice productions are in high trajectory from 1961 onwards, as can be seen in the graph also. Now, this was made possible by the efforts of the Rockefeller and Ford Foundations, and of course also along their Norman Borlaug. Now, aside from that, the establishments of the CIMMYT and ERI contributed immensely towards the development of modern varieties of wheat and rice, thus contributing to a higher agricultural output. At the local front, the agriculture, fishery, and forestry productions from 2003 to 2015 is far lower as compared to other sectors, for example, like industry and services. In fact, the agriculture sector only contributes around 9.7% share of the national GDP, despite the Philippines being classified as an agricultural country. Now, to worsen the scenario, in 2017, the agricultural sectors continues to hire like around 26% of the labor force, And this lead to a lower income in rural areas, which is then translated into a high poverty incidence. Now, in the Philippines, Mindanao has been dubbed as the food basket of the country. Our total land area is around 37,000 square miles, making Mindanao the second largest island in the Philippines, with large contiguous landmass, rich soil, abundant rainfall, and its locations that is located outside the typhoon belt makes agriculture's conducive activities in Mindanao. And in Mindanao also, most of the large investors have been investing in agriculture, which is translated into 40% of agricultural products that is supplied locally. And it also contributes like around 30% to the national trade. Now, this news clip that I got from Inquirer in 2016, which clearly indicates that there were at least 26 large firms that invest more than 1 billion pesos in Mindanao for agriculture alone. Now, while there has been a large-scale adoptions of technologies that lead to increasing agricultural productions, however, the growing populations and erratic climate change pose a new serious challenge to attain food sufficiency. Now, the pressing questions now shifted into how to push agricultural production curve outside when the conventional method seems to reach its saturation point. Now, this seemingly optimistic answer that I can give lies in exploiting germplasm variations using the modern platform of genomics. Genomics, on the other hand, refer to the study of an individual genome, that is the interactions between genes and the environments. Genomics as a technology platform has also allowed the systematic exploitations of the gene, which is also known as the blueprint of life. Genomics were also broadly classified into several distinct areas, some of which were listed here. The transcriptomics, which allow the interrogations of the RNA expression, the genotyping, which allows the investigations of variations in DNA sequence, and of course the bioinformatics, which allow the analysis of large biological data set. Now, so why engage in genomic research? Genomics has also allowed the determinations of complete nucleotide sequence of many organisms. And it also allows identifications of all of the structural and functionals of genes, which then allow the exploratory investigations of regulatory control of the genes. And the study of genomes of a crop has been made possible with the introductions of a new sequencing chemistry platform. To interrogate the DNA molecules in the sequencing workflow, the DNA samples will be processed to ensure a high quality samples were obtained. Subsequently, in the library preparations, we allow each samples to be uniquely barcoded during multiplexing. This allowed the batch sequencing run where multiple samples can be loaded at the same time in the sequencing machine. Then finally, the raw data that is generated in the form of FASTQ file can be interrogated using various computational tools. Depending also on the sequencing chemistry preparations, we can have RNA-seq, targeted-seq, whole genome sequence, as well as whole exome sequence. Generally, the processing of the raw FASTQ files follow the scheme. These steps were divided into 4 major steps: the quality control, trimming, filtering data, sequence assembly, and assembly validations. The QC steps gives a summary of the file and allow us to assess the data, how good your data is. The next is to trim the ends of each reads to remove the adapters and erroneous sequence, so it will not give a false result. Depending also on the objectives, we can pursue the genome assembly and validating the assembly result by running the assembly on several assembly software. Now, by overlapping the reads, contigs are assembled from short reads before scaffolding by large insert reads, and the remaining gaps are then filled. The scaffolding and the gap filling steps can be repeated several times until there is no contigs or no additional gaps are resolved before completions. Through this procedure, a draft genome can be built, but however, there are also some unfilled gaps that may still remain in the draft genome. Now, another way to process the high quality sequence right after QC are to look into the sequence polymorphism. Polymorphisms, now this test is usually done by invoking variant calling pipelines, which is composed of at least 10 steps. And these procedures reduces the full row sequence down to its few meaningful variants or the polymorphic sequence. Now, for this workflow, it is necessary to have the reference genome of your crop of interest generated first. Now, without this reference genomes, um, it is impossible for us to perform this variant calling pipeline. Generally, this test will map your row sequence to the reference sequence, and any base pair difference there are from that of the reference sequence, it will be called. And the final result of this workflow is the variant call format file, or the VCF files, which is actually can be processed further downstream. And the applications of these genomics tools into solving the key problems in the agricultural sectors give rise to what we currently know as agricultural genomics. These genomics technologies of the genomics platform offer a powerful platform to harness genetic gains in crops. By maximizing the utility of tools in genomics, we will be able to fully exploit the diversity of crops and animal germplasm, such as the high-yielding and disease-resistant traits. The agricultural genomics or agrigenomics itself has been evolved into several fields like bioinformatics and statistics, proteomics, metabolomics, functional genomics, as well as omics of biodiversity. And one of the major applications of genomics is in crop improvement. Indeed, the advances in the sequencing chemistry and its applications have led to the breakthroughs in this crop improvement. It was also important to accurately cataloging these resources by using genomic data, as it could provide a precise and usable information to plant breeders. Now, the sequencing results also expand the sequencing data in GenBanks and allow for a comparative analysis to provide a holistic view of your crop of interest. Another applications of genomics is in plant pathogen characterizations. The usual workflow in this initiative, or in this kind of research, is that it involves the genomic DNA extractions first, of which the samples are then undergoes an initial PCR. This is actually to generate a unique barcoded amplicons. Now, these multiplexing steps will allow for sequencing of several samples. The next is the barcoded amplicons are prepared for next generation sequencing. Now, and finally, the resulting sequencing reads are aligned to the reference database to identify the pathotypes in the sample. I would like to classify the tools that is widely used in agrigenomics into three main groups. And this is based on the natures of the data or the biological hierarchy that we are going to work on. So these levels are biodiversity, the plant bioinformatics or bioinformatics in general, as well as population genomics, a field that I usually actually, have a more experience with. So I think that there is a need to emphasize the importance of to preserve germplasms, as this constitute an important layer for a food secure nations, and it also serve as a reservoir for unique traits and genomic and genomics as technology will allows us to interrogate this germplasm and then try to look for a unique traits that that can assist and help the plants to adapt a different environmental stress. And this is also by accurately cataloging these resources and study of the variations of the genome of wild populations. The data that is generated can guide the crop improvement program. And the impact of climate variability, as well as climate change on agricultural productivity, will be likely to be a major constraint to achieve food secure nation status. And this makes the development of crop genotypes with resilience to, to climate change is an important strategy for food security and underscore the need to preserve our diverse germplasm. Another approach that has been very successful to investigate the complex microbial host interactions is the metagenomics. The metagenomics is usually used when we are studying microbial communities, when the culturing one microbes from another is next to impossible. Now, metagenomics has the potential contributions towards the study of the agriculturally important microbial populations, particularly in the soil, such as investigating the plant growth promoting bacteria. And the marker-assisted breeding has been widely used recently and become a standard in molecular breeding approach. Now, this approach uses DNA markers as indirect selection criteria for important traits in crop breeding. For example, markers that flank the target genes or closely linked to target genes can be designed, and this will minimize the number of backcross generations that it needs. And this is an example of marker-assisted backcrosses where the F1 generated were checked for true hybrids using DNA markers and then backcross with the respective recurrent parent to generate the backcross F1 seeds. And the subsequent generations will then be processed and handled. For the foreground selections for the genes or the QTLs will be carried using the gene based on the tightly linked markers or those that flank the gene of interest. And the selections for the lines with minimum linkage drag was done using both the proximals as well as distal markers or for the recombinant selection. Now, bioinformatics in a nutshell is highly reliant on the development of new chemistries and informatics platforms. The first generations of the sequencing platform could infer nucleotide identity using DNTPs, which is then visualized with electrophoresis. The fragments obtained are ranged between around 500 to 1,000 base pair fragments. The second generations of the sequencing platform uses parallelizations in its sequencing reactions. And while reads were shorter fragments than its first generation cousins, they are more cost effective on per basis and are also high throughput. The third generation's platforms, on the other hand, are capable of sequencing of tens of kilobytes base pair fragments on average. And in PGC Mindanao, we are using, we have 2 units of mean ion sequencer. So this is also the mean ion sequencer is an example, is one of the leading sequencing platform for the 3rd generation sequencing machine. So the sequencing platform are very dependent on the method applied, such as, for example, the power sequencing, sequencing by synthesis, ion semiconductors, single molecule sequencing, as well as nanopore sequencing. The main difference among these systems are on the length the sequence reads, and the unique error model that they apply to correct them, and the operational, as well as the operation cost to operate these machines. Now, these differences affect how the reads are utilized in bioinformatics analysis. And depends on the ultimate goal of a particular research that one may choose. You may choose a different sequencing platform that is also dependent on how would you apply it in your research. Now, the plant genomes have contributed many valuable materials for crop research in modern molecular genomics era. Now, the availability of complete genome sequences as well as the explosions of sequence data is leading to an urgent need for a well-cataloged and annotated DNA sequence databases. The largest and most well-known of these sequence databases are Genbank, DEMBL, and the DNA Data Bank of Japan. And here are some of the most widely used resources. And since I work mostly in crops in Gramineae family, I occasionally visited rice genome projects and Gramineae websites. For comparative genomics approach, it has been used to predict functions of many unknown genes by studying the significant differences and similarities among species. However, in order to perform comparative analysis, these genes are required to appear in the available database of orthologs evolved from the same ancestors. Not only by comparing within plant species, comparative genomes between plants and distantly related prokaryotes can also greatly presume the genes functionally associated with. As can be seen, the enormous amount of data for plants rapidly increases, and there is a need to develop new tools and update current ones. Strategies to manage and analyze this tremendous data has been also urgently needed. Aside from this, the reference genome sequence with basic annotations provided by current comparative genomic databases is also a foundation. For example, the current sources still needs to be continuously populated with specific biological data, such as the plant epigenetic decorations and gene expressions under varying conditions of environments, development stages, and tissue types in order to get better detailed genome maps. And the phylogenomics, on the other hand, has an advantage over others. The plant phylogenomics, on the other hand, has an advantage over other species, which is the ability to identify hundreds of low copy number of nuclear genes. Hence, it makes it easy to study the molecular systematics and evolutionary biologies. The current approaches of the NGS, or the next generation sequencing, also provide plant phylogenomics research useful information about plant genome diversity, such as the nature and frequency of genome duplications among diversity of plant lineages. There are two important goals in phylogenomics research aims to accomplish. First is to discover the evolutionary patterns among plant species using nuclear genome information. And the second is to derive the new hypothesis for the unknown functions of plant genes associated to major divergence events in the evolutions of plant species. And the genomic data give more advantages in the evolutionary study by using a set of orthologs from genomic sequence. And this should be done in a phylogenetic context. And this is to detect the hypothesis for the genes and biological processes. However, the main problems for the phylogenomics come from how to handle the large scale of genomic data and the proper way to avoid systematic mishandling or biased assumptions. However, currently there are new methods and tools that have been developed gradually to overcome these limitations. For instance, de novo assembly of short reads dramatically improves gene coverage by non-redundant and non-chimeric transcripts are optimized for downstream analysis. Another protocol is called the hybrid-seq. This combines the target enrichment of low-copy nuclear exons in flanking regions, as well as the genome skimming of high-copy repeats and organelle genomes. And this is to produce a genome scale data sets. The genome-wide association study is another approach used to associate a specific genetic variations with particular traits. Now, this involves scanning of genomes from many different individuals and looking for the genetic markers that can be used to predict the presence of a certain traits. In such a study, distributions of SNPs is determined in hundreds or even thousands of individuals. Now, and by tallying which SNPs co-occur with traits, researchers can make a statistical estimate on the level of increased risk associated with each SNPs. I would like also to share some of the work that I did, particularly in the identifications of the novel PI29 alleles from the 3000 rice germplasm. In this work, I used the available core SNPs data set of 539 land races. Then I conduct a pathotyping screening and challenge against known as well as novel isolates, and then perform the associations analysis. I usually perform this using 3 association models. That here I just only show the third associations model, which is the associations accounts the kinship relationships among the sampled individuals. Now, the figures in the upper left show that the Manhattan plot and the lower left shows the QQ plot. It can be seen from the Manhattan plot that the peak clearly overcome both the genome-wide significance threshold and the suggestive threshold values in chromosome 6. Now, it is also interesting to note that the peak also lies in the regions that has been previously confirmed to contain PI2, PI9 gene. And the QQ plot that is used here is to assist population structures. The presence of population structures can lead to false results, and it is very important to take this into account during performing association studies. I also compared the allele frequencies of the resistant populations and the susceptible populations by retrieving 12.5 kb windows upstream of the GWAS peak and the 12.5 kb windows downstream of the GWAS peak for a total of 25 kb window. Now, these figures here show that the resistant allele tend to contain more significantly more AG allele and susceptible line to have contain more of the TC variants. For the rest of the variants, there seems to be have a smaller effect in that region. Also, to visually inspect the haplotype patterns in the 25 kb region surrounding the GWAS peak, there are 7 populations. I also constructed this heat map. Here, I'm using this 7 subpopulations of rice. With indica subgroup showed a higher number of representative. The SNPs positions occupies the central part of the figure, and the disease states occupies at the lower graph, lower bar graph. And as can be seen from the graph, there are 2 major resistance haplotypes that tend to distinctly linked and cluster together, that is the A and B. It is also interesting to note that the majority of the tropical subgroup tend to harbor disease-resistant haplotype, as can be seen by the orange at the subpopulation graph bar. Another very promising approach is the genomic selections, where in exploiting molecular genetic markers, it could design novel breeding programs and eventually develop markers-based models for genetic evaluation. In breeding crops, it provides an opportunities to increase genetic gain of complex traits per unit cost, per unit time and cost. And the cost-benefit balance was an important consideration for the genomic selections to work in croplands. And the successful and effective implementations in crops depends largely on the availability of genome-wide high-throughput data, cost-effective and flexible markers, and having low ascertainment bias, and also, of course, suitable for suitably large population size. Here in the genomic selections, here the genomic selections is made up of 2 populations: the training as well as the breeding populations. The training populations were both contain both genotypes and phenotypes. This data will be used to train computer system to create a statistical model that could relate the genotypes and phenotypes. And the trained computer system is then would then predict the genetic estimated breeding values of the breeding populations. And as in recapping the talk of this afternoon, I would like to say that advances in the innovative platforms like genomics can be utilized to benefit society. Despite it is relatively expensive to engage in this kind of research, compared to conventional approach, it pays very well in the long run. For take-home message, I would like to share some insights from reading these books. This is book written by Yuval Noah Harari. So while the author paints a gloomy historical anecdotal, it is still worthwhile to say that issues on securing food to feed the growing mouth has been with us for nearly as 10,000 years. And it has also been the foundation, and I would also like to underscore that the agriculture has also been the foundations of all modern civilizations. They are facing with a different constraint, they are facing with a different problem, so they approach those problems differently from us. Now, we are currently in the 21st centuries, and it is also important to highlight the availability of modern platform such as those that is available in agrigenomics, can actually greatly address our concerns of feeding our people. So before I end, I would like to acknowledge my PGC team for all the support given in this first lecture series, first bioinformatics lecture series.
Thank you very much, John, for providing us a glimpse on the widely used genomic tools in agriculture in efforts to boost agricultural production. May we request you to join us in this virtual screen? Hi, John.
Hi, Joe.
So we have a couple of questions here in our Slido app, and we have 20 minutes for you to answer this.
Okay.
So I would also like to invite our participants to just send in your question, and you can also upvote here some of the questions which are already asked. So shall we start?
Sure.
So the very first question here is from an anonymous participant. Which is more important plant trait, high yielding or climate resilient? Or can both be developed in a crop? Yeah.
Okay, so for me, I would say both are important. Both traits are important, but I think the concern here lies if we are going to breed for a high yielding variety, will that genes for the high yielding genes will actually have an effect towards the to the crops for its climate resiliency traits. So it will also depends on those mode of inheritance for that certain crops.
So there is an follow-up. How long do you think this will usually take if you both combine the traits?
Okay, so for the breeding, for the breeding of these crops for these certain traits, I would say it will last like around 7 to 12 years. That's why we usually do population genomics. We do GWAS to actually shorten the breeding um, the, the breeding cycles to like approximately around 3 to 6 years.
Right. So here is another, the most voted question. How are these omics tools currently being used in the country for agriculture? Are they specific products or projects you can cite as examples?
Yes. For in the Philippines, actually, most of these agricultural genomics projects is actually been being piloted in UPLB as well as in Erie. So in Erie, we have this 3,000 rice genome projects, and the availability of the database makes it very easy for us to identify which genes actually control for certain traits. Yeah, example of that also is the PI2, PI9, which I'm actually also working on for my thesis.
All right, thank you, John. So here's another question. So what do you think are or will be the trends in the future in genomic research, especially in its biotech applications? Thank you. This is from Hansley Rodriguez.
Okay. For me personally, I think in the future really lies on the genome editing. The bioinformatics as well, the sequence analysis as well as the genomics that we are dealing a large data set, it will provide the platform for a more advanced tools to be developed.
Okay. So here's another one. So we have several questions here. And for the PGC related question, we will ask later. So we have 20 minutes for it.
Okay.
Okay, so the question is from an anonymous participant. Does the Philippines have the capacity to perform high throughput genotyping to develop dense SNP marker panels through GBS. It's moving. Wait lang. Ano
For
yun?
yeah, for the initial,
Particularly for GWAS.
yes, we have that. We can perform actually whole genome sequence, and then we're going to map that against the reference, the available reference genome. Then we will be able to identify the variants. The answer is yes.
Okay. Yes. So there is also a question regarding specification. May I ask what is the recommended computer system, specifications, OS, etc. for studying bioinformatics? I'm a graduate student new to this field.
Okay. For me personally, I would suggest an operating system based on Ubuntu or Linux platform. And then the minimum specifications for the RAM should be like at least 4 GB of RAM. And then yeah, at least 4 GB of RAM. But currently you can have currently the specifications for the workstations now will be like around 8 GB of RAM. But 4 GB is generally okay. But you really need to use an operating system based on Ubuntu or the Linux platform, because Windows, if you are using Windows to analyze the biological data, yeah, that is okay. However, because of the large of the data set, and then also Windows, when you open Windows, there are more memory that it consumes than it should be allocated to more space for the data analysis. So I would say that is the minimum requirements.
So there's also a question regarding program
Okay.
from an anonymous participant. Hi, John. Great talk. I'd like to know, do you have recommendations on program or software to use for the detection of transposable elements or retrotransposons?
Oh, okay. For this, I have to say that I did not work on these untransposable elements. However, I think we can check this on the NCBI, because I think when we identify these genes, there is an annotations that will particularly there will be an annotations of the SNPs or the variants that would tag it whether it is a transposable elements or a retrotransposons.
Okay. So from an anonymous participant, how many plant samples or genotypes are recommended when doing GWAS? How do you account for the environmental effects?
Okay, so that is one of the problem also. It really come for the population size, right? So for GWAS, it is recommended to really work on the natural populations, and then hundreds it's preferable, at least 100. In my case, I work on GWAS at least on 539 lines, so I have somehow a good sample size, and then it is also only available in ERI database, so I'm lucky for that. I'm very lucky. I'm very lucky with that. If you really need to do for GWAS, so you really need to have at least 100 samples, 100 individuals to be involved. It also depends on the power to detect that variance. Like for example, if there is a minimum like around 80% power to detect the variance.
So the minimum is 100.
Yes.
Okay, that's a lot, no? Okay, another question from an anonymous participant. Aside from rice, are there any progress on other grains or vegetables in using these advanced tools?
What is that, Joe? Can you please repeat that?
So aside from rice,
Okay.
so do you know any other crops or other vegetables using this advanced tools,
I
the
think
progress?
I think in tomato they do similar. They have also similar. They also using this similar platform. As to the other agronomic crops, like for example corn, I would say there is an effort to do that, but corn is polyploid, then it's very difficult. For example, if you want to perform the GWAS or the genomic selections, because of its ploidy level is different, and you have to really account that.
Right. So yeah, we still have a lot more. Another question from Jay Sanchez. Bioinformatics is not instituted in the curriculum of bio program. There is a niche because of the pricey. It's moving again.
Yes,
Where is
that's
it?
true.
HPC and other instruments. How do you see it in the next 5 years?
Okay. Well, in the future, the future will really be dependent on, or will be very reliant on, the availability of the data for us to work on. So I think in the future, there will be a program that that will incorporate bioinformatics in their curriculum. Because, for example, in UPLB, there is ongoing talk actually to have a bioinformatics program, as far as I know.
Okay, so there is a future for that.
Yes.
So actually, there is a follow-up question regarding the minimum number
Okay. Okay.
from anonymous friend. In GWAS, the minimum is 100 varieties or genotypes. Then how many individuals are needed to account for the variation within varieties?
Account variations within varieties. Okay. So this is the this is the questions of to how to address the population structure, I think. So to address this, we have to have at least as uniform number of individuals per each genotype. Yeah. So for example, if you have, let's say, for example, because I'm working on rice, so if you're working in subpopulations Tropica or Indica, you have at least to have 50-50 each subpopulation.
Okay. So you can still have your follow-up questions later. And so there's another question. How does the pandemic affect the bioinformatics field? Do you experience any limitations in research?
When it comes to bioinformatics, no. Actually, that is the trend now because we cannot do laboratory. However, we are very lucky in PGC because we are given this privilege to conduct this. We are very privileged to have this wet lab research still ongoing. However, yeah, that is the trend now because of the pandemic. Because that's it.
Yes, there's a specific question, John,
Okay.
from JT. How can genomics contribute to solve the root cause of climate change?
Okay, so you see,
wait lang.
Sige, Jo.
Change which influence the yield resistance of the usual or current cultivars.
Where is that?
JT,
How
from
genomics
JT.
solve the root cause of the climate change? Okay. Which influence the yield resistance of the usual current based cultivar. How can genomics contribute to solve the root cause? You see, we cannot. I don't think that the genomics can really could really like totally solve the problems. But the genomics is one of the tools, one of the many tools that we can use to address the climate change. Because of that, I think we have to look into a bigger bigger perspective now. Because of, for example, if you have a plant that is not that is very prone to the climate change effect, the problem there is that we tend to use for example, more chemical treatment, so that crops will not experience any down any downgrade in yield. Then I think we really have to exploit on that the genotypes of those plant. Yeah.
Okay, so we still have a lot more questions here. Are you still okay, Jen?
Dami. What did you
Or you have seen the questions, right? You can pick the questions.
Yes.
So
Okay.
it's
Okay.
okay.
Okay. I will just
All
go
right,
through this.
right. Okay.
Okay. So how do you think
So
the genome?
there's
Sige,
a
go, Joe.
question here. What is your take in the commercialization of golden rice? Would it benefit the local rice farmers?
My answer to that is definite yes. I work with Erie. When I work with Erie, the principal investigator is just only right by my office, so I know what's going on there. So I think they are doing a great job in that aspect.
All right. And it's nutritious, no? There's another one upvoted. What can you infer from the GBS data? How can you use this for crop improvement?
Okay, so when we use the genotype by sequencing, we are actually reducing, instead of using the whole genome, we are just only look for the variations for specific SNPs or for specific what's this variants. So in that case, it becomes more cost effective. And then when we look for the variations in that, we can actually correlate them or associate them to a certain traits. Then it's much cheaper as compared to whole genome sequence. And may I pick also this one question, Joe?
Okay, your favorite.
Can we perform GWAS if no reference sequence available for our species? The
Yeah,
answer
yes.
is probably not, because if there is no reference sequence, so we could the approach usually what we can usually do is actually to look for a homologous sequence or homologous reference genome. If there is none, so we have to really have a good reference sequence. So if you will notice in the reference sequence, there is several versions that's because they are actually reanalyzing the same data set again and again to come up with a better reference sequence. That's why whenever we will going to map again, we will come up with a new SNPs, with a new variants that can be called.
So we need the reference.
Yes.
Okay, so
okay, ito pa isa.
do we have, what's this?
Is it actually a good idea to major or minor in bioinformatics or just take a course about it? Thank you. Okay, agri-biotechnology graduate. I think this is from Biska ata to. But anyway, let me answer this then. For me, bioinformatics is an interdisciplinary field. Then you can actually be the one that will really have an advantage to work in the bioinformatics is those who have a strong computational background, like math, statistics, computational chem, physics. Biology will be very helpful, but you really need to exert more effort to learn this very hardcore field. So that's why in our project staff here in UP, in PGC Mindanao, most the one that lead this is actually our faculty from the math division. Okay.
So there's a question, John. Are there any
Yeah.
institutions or organizations here in the Philippines that provide bioinformatics consultations for free or at a low cost?
PGC Mindanao, I guess. But we are not going to offer it not free, but the cost effective, yes.
Minimum.
Yes.
Okay, so there's a question regarding the whether you use Ubuntu or Mac OS for bioinformatics and
Okay.
compatible software for Mac.
Yeah. I'm not a Mac user. I am an Ubuntu user. So I have my boss before. He's using Mac, but he told me to run all of the analysis in Ubuntu because I think the Mac cannot carry some of the analysis that is quite heavy. Okay. My question to favorite then. Do you have current projects in rubber genomics? The answer is no, but we are very interested to that. Very much interested.
All right, so there's also a comment here from our chat box from Miss Vivian Calambro. Yes, golden rice will not only benefit the farmers, it will also benefit people in countries that have high incidence of vitamin A
Vitamin A
deficiency.
yes.
True, that's true. Thank you for that comment, ma'am. Okay, so in Slido, we also have other question. How long does it usually take to accomplish bioinformatic, where is it? Analysis of a single genome?
A single genome depends. Depends on the depends on your objective. But for example, if you want to conduct for the whole genome for the genome assembly, I did a run on Adlai. Then it would take me like around 8 hours. The file size for that is around 20 gigabytes, and then yeah, I have to wait it at least around 8 hours maybe. Then I were able to have the result. But I ran it in HPC. Now, for that HPC, I really use a very powerful resources because here in PGC Mindanao, we have a very powerful HPC. It's actually housed in UP Diliman as of now, but we are planning to have it here so that our researcher in Mindanao can use it. And then after the genome assembly, after 8 hours, then you are going to do variant call for the SNPs analysis or to look for the variations in there. Then you will have like another, let's say only one genome, so it could probably run like around 12, around 8 hours again. Then you have also the more downstream analysis, for example, genome annotations. You're going to look what is there inside. So how many untranslated, what is the retrotransposons, and etc. So probably for a good result, you can have like around, I would say like around 2 to 3 months.
Okay. Thanks, John. So there's a very specific question here. Primers recommended for orchid species and Papapedilum species
Okay, I think we can use
for
the
identification. So there's the universal primers, right? Okay, John.
yeah,
please.
that's true. You just only use a universal primers. I think Joe, you can answer that.
Yeah, we have markers for DNA
Yeah,
barcoding for plants like RBCL, MATK, and what else, John? Yeah, that's for short segments. Anyway, if you really want to know more about this primer, we can look for some literature. No?
yes, that's true.
Okay.
Ito pa, do you plan to engage in projects related to forage pre-development for our livestock sector? The answer is yes, very much. Please do contact us.
Yes. So someone's asking for advice. Is it actually a good idea to major or minor in bioinformatics or just take a course about it? Thank you. I'm agri-biotech graduate.
Yeah, I think I answered that. The answer is agri-tech graduate, right? Yeah, you can minor, but I would suggest that you minor like either statistics or computer science, but I would prefer statistics. Because data science, bioinformatics is just only an applications of this very of the traditional field. And you will not see the clear lines or the clear boundaries that eventually separate them.
Okay. There's a question. How do you think the general Filipino consumers will react or welcome these GMO crops that aim to improve, if not completely replace our existing crops?
Well, okay. I think this will be very dependent on the how are we going to educate the general populations on the on this GMO. So it will still be dependent in how we're going to publicize this. Whether will be definitely this is good based on our study. However, how are we going to convince them? Because generally in the Philippines, I have to say that we are not yet that scientifically literate enough.
Yes, can you emphasize more on the difference between reference-based and de novo assembly?
Okay. If we have a reference-based genome assembly, as you have already existence, you already have an existing reference-based genome, and then you're just only going to map them, similar with the variant call. However, it will give you a different result. When we say de novo, we are going to use the only that sequence of an organisms, and then you're going to establish. They are going to construct that whole genome based on a specific algorithm that is involved.
All right. So I think we still have more questions here, but these questions will still remain in this Slido link. So John will reply in this link. Just visit your questions here to see the answers.
Okay.
Okay, so yeah, but before that, we still have one last question.
Does PGC Mindanao?
Yeah, about PGC Mindanao, what
internship program for undergraduate students for the next academic years. The answer to this, I have also questions. Does it on the bioinformatics? A bioinformatics online, yes, we do catered for the online internship. However, for the hands-on or face-to-face, I think it depends. It depends on the IATF guidelines.
All right. Thank you so much, John, for that engaging.
What tips or advices can you give individuals new to bioinformatics? I would say read, and then try to solve as many problems as you can. Accept whatever, when people consult you on a problem on the sequence, accept them without any fee, because it means that you're handling a different set of problems, a different set of objectives.
Thank you, John. Yeah, our 20 minutes is up. Okay. All right, so yes, don't worry, all your questions will be answered by John in this Slido, and even in this chat box. We'll also keep those questions and relate to you the answers later. All right, so again, thank you for this engaging Q&A, John.
Yes, thanks, Joe. And also,
I would like to turn you over to Ma'am Chini Gamot.
Hello. Thank you, Jan and Joan, for that very engaging question and our answer portion or session in this webinar. So I'd also like to add, because I think one of the, or a couple of the participants inquired about a bioinformatics program. I'm actually one of the sort of core group in developing a master's and a PhD bioinformatics program. This is headed by UP Diliman, the Department of Computer Science, but it is being assisted by all the UP units in the UP system. So we have currently drafted the operational guidelines and all of the courseworks and needed deliverables for graduate studies in bioinformatics. So to those who are very much interested in enrolling, you might be able to enroll either end of next year or maybe early 2023. So antabayanan po ninyo yan. UP will be offering MS and PhD in bioinformatics, and it will be a collaboration of all the UP units in the UP system. Okay, so in this juncture, again, as much as we would like Jan DIVINA to answer all your questions, we only have a short amount of time for him to answer it. But don't worry, you may contact Jan, or you may relay your questions to the PGC Mindanao email address, and we will assure you that we will send your questions to Jan DIVINA so that he can also answer your questions through email. Okay, at this point, we would like to give our appreciation to our moderator and our speaker. But before that, I would like to request for our moderator and speaker to turn on their cameras. And aside from this one, I also would like to call on our director, the program director of PGC Mindanao, Dr. Morao, to turn on her camera as well. Okay, do we have Ma'am Lyre na po ba?
Sorry, cannot turn on.
It's okay, ma'am. No worries po, ma'am. No worries po. Okay. So let me read the citation. PGC Mindanao, College of Science and Mathematics, University of the Philippines, Mindanao, gives the Certificate of Appreciation to Mr. John B. Divina for being a resource speaker on genomics and agriculture, toolset of agricultural genomics during the first lecture of the Bioinformatics lecture series held on October 1, 2021. Signed by Dr. Lyre Ann Imarao, Program Director of PGC Mindanao. Thank you, Jan. Also, we would like to give this Certificate of Appreciation to Ms. Joan T. Acaso for being the moderator during the open forum of the talk Genomics and Agriculture, Toolset and Agricultural Genomics of the Bioinformatics lecture series held on October 1, 2021. Signed, Dr. Lyre Ann Imarao, Program Director of PGC Mindanao. Thank you, Ms. Joan.
My pleasure. Thank you.
Okay, so thank you again, Jan and Joan. But I would like to request both of you to turn on your cameras muna because just like any Filipino-Pinoy gathering, conference event, we have to take a photo. Okay, so this time it's a photo opportunity. So at this point, we would like our staff to do the picture taking. So one of our staff will give us a cue for picture taking. So Jan and Miss Joan, just give us your best smile siguro. Okay? Sige, so smile na lang. Okay, okay na po ba? Okay, so we also would like to request all of the organizers of this first talk in Bioinformatics lecture series. I would like to call on all of the staff from the Research and Development Division of PGC Mindanao and as well as the staff of the Bioinformatics Division of PGC Mindanao. These staff were instrumental in preparing this Bioinformatics lecture series. And even if you are not from those two divisions, if you are part of the panel members here in Zoom, you may turn on your cameras, okay, for a photo opportunity. And of course, again, one of our staff will cue us for a picture taking. Okay, so we have in here Dr. Ugis, Dr. Tolentino. Who else are turning on their cameras?
Jim, nakalaking video ko.
Okay, so we would like to ask our technical team to allow Ma'am Lyre to turn on her video.
Hindi din ako maka-open, James.
Oh, okay. Ano po natin, Ma'am, i-ask po natin yung technical team natin. Hopefully, they will be able to resolve the problem. Okay. So I think Ma'am Anna is also requesting for a video turning on of her camera. Ayan, ayan na. So we have Dr. Novero as well. Who else? Ayan, so we have Dr. Morao, our program director.
Dr. Alela.
Dr. Dicadis, can you turn on your camera? Or are you restricted as well? Because we can resolve that. Our technical team are experts on...
Wala akong lipstick, okay lang.
Okay, ma'am. This is Okay, ma'am. Thank you for informing us. Okay, so are we good? How about Paul? Do you want to join us? Do you have a camera on your machine? Okay, opt out. Okay, we respect that, of course. So I think this is a group who wants to be in our on our photo opportunity. Okay, so everyone, tidy yourself up and then give us your best smile. Okay, sige, who's going to take the photo? Ako na lang. I'm going to take the photo while our technical team are positioning themselves in the camera. I can actually do the photo ops. Okay, sige. So everyone, please give us your best smile. Dr. Dicadis is here with us. Sige, smile pa. Okay, thank you very much. Thank you, everyone. Okay, you may want to leave your cameras open, or if your camera is shy, then you can turn it off. Okay, of course, we would like to hear, aside from our speakers and our moderator, we would like to hear from our participants on how we did in this activity. So on the chat box, our staff will be providing you the link for the evaluation form. Please do evaluate our activity so that we will be able to improve on what we have missed in today's lecture series. Okay, so we hope that you are able to evaluate us within the day or maybe until tomorrow, because after that we will be closing the link. Okay. Okay, so for future events and to know more about the services that we offer, we would like you to visit the PGC Mindanao website or our various social media accounts. You may also contact us through the number shown, or you may email us through pgc.upmin@up.edu.ph. Our activity today is just the beginning of an exciting lecture series on bioinformatics. On October 29, we will be having Professor Jean-Baptiste Cazier to talk about the UK efforts in the coronavirus cancer monitoring project. Our speaker will be talking about XAI, or explainable AI, as applied on data gathered across the UK of cancer patients with COVID-19. Also, on November 2, Professor Janusz Kristonvizi will be gracing us with a talk on bioimage analysis. The talk will be about using high-content screening, a bioimage analysis technology, to identify small molecule compounds that effectively control autophagy in cancer cells. We hope that you could join us again on those days. Again, on behalf of the R&D and the Bioinformatics Division, I'd like to thank you for attending the very first talk on the Bioinformatics lecture series of PGC Mindanao. We hope that you have a nice evening ahead of you. And stay safe, everybody. Bye!
Jan, sumigaw ka. Parang sumigaw si Jan.